Connie, if you start the recording, she has, so you're ready to go, Isabella.
Hi, I'm Isabella and I am working on TOPOCAN, a novel histopathological system for clinical prediction with topology and spatial genome-informed deep learning. Cancer is the leading cause of death worldwide. Breast cancer is the most prevalent cancer with 2.5 million cases in 2021 alone. Thus, improving early diagnosis, prognosis prediction, and targeted treatments is crucial. Currently, pathologists rely on experience-based qualitative image cues when examining histopathological tissue samples, which can't account for cancer complexity and heterogeneity. To better understand the development of cancer, we must first understand the tumor microenvironment, or TME. It regulates essential tumor survival and promotion, invasiveness and growth, resistance, and clinical outcomes. But there's one key obstacle to our understanding: tissue heterogeneity. The TME is made up of many different types of cells, making it difficult to pinpoint interactions of cancer cells. To understand cancer regulation, we must first comprehensively characterize the TME. Current transcriptomic techniques, such as RNA sequencing, can reveal cell identity and functionality, but not the context of the cell's neighborhood and how cells are arranged. This missing spatial context reveals key information about cell-cell signaling and how tumor cells are regulated and activated. On the other hand, imaging data can reveal spatial relationships and cell arrangements, but not the identity of different cell types. So how can we combine these two to gain a complete understanding of the TME? The advent of novel spatial transcriptomics techniques, or ST, has completely revolutionized biomedical research by enabling the characterization of not just cell types, but also their spatial organization. Each spot in the ST sample has a unique barcode for identification, so it can be mapped back to its original location. At every spot, we can obtain localized image features and gene expression vectors. Now, how can we connect these two types of data? This connection is crucial to understanding the impact of tissue architecture and cell-cell interactions on disease development and progression. Before this is possible, there are a few limitations that must be addressed. There's no existing systematic method to interpret and quantify these significant cell arrangements, limiting their ability to be analyzed and clinically applied. There is currently no known method for linking imaging features to transcriptomic features. And finally, there is no way to apply what we learn from a spatial transcriptomic data to a general clinical setting where SE data is not available. To address these limitations, I designed a novel system called TOPOCAN with three main objectives. One, characterize and quantify the spatial arrangement of cells, otherwise known as cell topology. Link localized gene expression with the novel spatial characterization of cell topology, and finally apply localized expression and topology for more accurate, specific, and immunologically informed clinical prediction. To characterize and quantify cell topology, image topological features are novelly extracted through a topological data analysis technique known as persistent homology. These image topological features, otherwise denoted as ITFs, characterize and quantify significant spatial arrangements. Here, the extracted ITFs represent ring arrangement of cells. 700 ITFs were extracted and can be interpreted for clinical predictions. This project has two main stages to achieve the objectives. In stage one, I directly linked topology and spatial gene expression through a topogene dictionary that maps them to each other. To the best of my knowledge, this is the first study that directly links topology features from cancer histology to gene expression from spatial transcriptomics. With the dictionary, I select the most prominent image topological features correlate with oncogenic genes as biomarkers and potential therapeutic targets. In stage two, topology and spatial gene features are effectively applied to clinical applications. In phase one, topology is exploited in a novel deep learning model to predict localized and spatial gene expression from patient images. In phase two, the combined power of topology and spatial gene information is applied to clinical prediction through genome and spatially informed deep learning. In stage one, I constructed the first topogene dictionary to directly correlate prominent oncogenic genes with topology features to gain a complete understanding of the TME. First, a Pearson correlation matrix was calculated between the gene expression matrix and topological feature matrix, which contains the enrichment of all 700 extracted ITFs. The most highly correlated genes and ITFs were identified and linked to create the dictionary. For each ITF, the top 50 positively and negatively correlated genes were selected, and vice versa for each gene. The dictionary reveals how gene expression impacts the spatial arrangement of cells. The dictionary identified the gene PFN1 and ITF122 as highly correlated, and their enrichment diagram shows that they are indeed nearly identically distributed enriched in the tissue sample. With the dictionary, I can select prominent input topological features that are highly correlated with oncogenic genes as biomarkers. Specifically, I identified ITFs that represent the interaction between tumor cells and the immune system, which is crucial for developing cancer treatments that help the immune system detect and kill tumor cells. In their interactions, cancer cells present MHC1 complexes, which highly express HLA genes. CD8 plus T cells, which is a type of immune cell, targets cancer cells by binding onto their MHC1 complexes with T cell receptors that express TCR genes. Therefore, ITFs positively correlated with both HLA genes and TCR genes are selected as potential representations of the interactions between the immune system and tumor. 7 genes and 26 ITFs associated with immune signaling were identified, and the correlation matrix is visualized here. The signaling genes included HLA genes and TCR genes. ITFs 104 through 128 are selected as novel potential topological markers, collectively referred to as tumor immune ITF markers. Through a functional annotation, these tumor immune ITFs were found to be highly enriched in important signaling pathways, including the cytokine-mediated signaling pathway and the antigen presentation by MHC class I, which is how immune cells identify and kill tumor cells. Additionally, they were enriched in the interferon gamma-mediated signaling pathway and cellular response to tumor necrosis factor, which are important regulators of the immune defense response against cancer. Additionally, ITFs 121 and 122, which were a part of the tumor immune markers, were visualized here. They are present at the border between immune tissue and tumor tissue. These pathways and visualizations suggest that ITFs represent the interaction between tumor cells and immune cells and can serve as potential targets for immunotherapies such as CAR T therapy or prognostic indicators. With the Topo Gene Dictionary, different types of novel topological biomarkers can be identified. For the first time, we can identify specific, quantifiable image targets to diagnose a patient, predict survival and treatment response, and more from the patient alone without genetic testing, increasing accuracy and reducing costs. In phase 1 of my stage 2, I performed spatial gene prediction through topology. With a significant correlation between localized extracted ITFs and gene expression established through the Topo Gene Dictionary, how can we now exploit this connection to make clinical predictions? To do so, I built a deep learning model to apply topology and gene information to predict localized and spatial gene expression patterns from image samples, which can be applied to real clinical studies where only imaging data is available. I introduced MIT-DCCN, a multi-input topology deep convolutional neural network, which improves prediction performance by exploiting both image and topology features. It consists of a multi-input network with convolutional layers based on ResNet 18 for image data and MLP neural network layers to input topological features. Both types of features are concatenated just before the linear activation and fully connected layers for prediction. Rather than only relying on image features like current models, MIT-DCCN is capable of making more precise predictions by leveraging the link between topology and gene expression. MIT-DCCN's performance was compared to a baseline CNN model trained only on image features. Both models predicted the expression of 3 important signaling genes, HLA-E, HLA-SC, and HLA-F. MIT-DCCN outperformed the baseline for all 3 gene labels, demonstrating that topology can be a powerful predictor of gene expression. MIT-DCCN can now be applied to new patient samples to extract ITFs and predict spatial gene expression, unlocking a novel avenue for improving clinical prediction. In phase 2, the results obtained through SC analysis are applied to clinical outcome predictions. Being able to predict the spatial gene expression of a slide using topology features, the features extracted through MIT-DCCN are genome and topology informed. They accurately capture and represent the topological and genomic information of a slide. For clinical application, these topological and genomic informed features were input into an STMIL prediction pipeline, which applies all significant findings from this study to the clinical prediction on TCGA breast cancer dataset for validation. First, TCGA slides are broken into patches. Then, features are extracted from the patches using the MIT-DCCN. These extracted features from patches were then input into an MIL framework to aggregate into a whole slide prediction label. SCMIL's performance was compared to a baseline MIL model trained only on image features. Both models were applied to tumor detection, tumor subtyping, and survival prediction. As shown, SCMIL outperformed the baseline MIL model for all clinical outcome predictions, achieving an accuracy of 95.7 for tumor detection, 92.5 for tumor subtyping, and 91.7 for survival prediction. Topology and spatial genome-informed learning greatly enhanced clinical prediction in new patient cohorts, indicating the significance and contributions of this study. Finally, TOPOCAN was integrated and developed into a user diagnosis and visualization application system, which is available for use by pathologists and doctors for clinical predictions and is accessible, efficient, and powerful. As shown, an input feature is first selected. The system then outputs the SCMIL predicted diagnoses. And then in this panel, ITFs of the user's choice can be visualized and their enrichment can be visualized to identify specific spots where the ITFs are highly enriched. This is TOPOCAN. There are four main contributions of the study. First, a topogene dictionary that links cell topology with gene expression for the first time. Tumor immune ITF biomarkers that reveal tumor immune interaction. The MIT DCCN model for spatial gene expression prediction on new image samples. And finally, the SCMIL model that integrates topology and gene-informed learning for clinical outcome prediction. TOPOCAN fundamentally advances spatial transcriptomics research. It links topology and spatial information to understand tumor heterogeneity. The power of TOPOCAN is that it is a generalizable framework. It can harness the power of SC and topology for any cancer and any clinical application, including diagnosis, treatment response, and more. In the future, this work will be expanded and validated on other types of cancers to improve the interpretation of these topological feature biomarkers in clinical settings. Finally, the image features extracted via the CNN will be further refined through advanced vision transformer models, and more advanced neural network architectures will be explored to further increase performance. This is TOPOCAN, the future of personalized healthcare. Thank you.